In 1843, a Swiss-American naturalist named Louis Agassiz published a description of a set of fossilized teeth that had been turning up in limestone deposits across Europe for centuries. Medieval scholars had called them glossopetrae, tongue stones, believing they were the petrified tongues of dragons or serpents. Agassiz recognized them for what they were: shark teeth, enormous ones, belonging to an extinct species. He gave the animal a name that placed it in the same genus as the great white shark, Carcharodon Megalodon. The name stuck for 150 years. The classification shaped everything that followed: the behavioral models, the size estimates, the extinction theories, the gift shop teeth. It was also wrong. The great white shark is one of the most studied predators alive. Carcharodon carcharias has been observed, tagged, sequenced, and analyzed in detail for decades, and researchers know its phylogenetic position with considerable precision. The great white sits in the family Lamnidae, and its closest living relative is the mako shark. Agassiz had assumed that because Megalodon's teeth looked broadly similar to great white teeth, large, triangular, serrated, the two animals must be closely related. In paleontology, tooth shape has historically been the primary tool for classifying extinct species. When the only thing you have is a tooth, the tooth becomes everything. What decades of tooth analysis slowly revealed was that the resemblance between Megalodon and the great white was shallower than it looked. The enamel microstructure of Megalodon teeth differs from great white teeth in ways visible under a scanning electron microscope. The root morphology is different. The overall geometry of the crown, when measured carefully, diverges in ways that parallel evolution. Convergence on a similar shape through independent lineages explains better than shared ancestry does. By the 1990s, researchers including Shelton Applegate and Applegate Espinosa-Arrubarrena had argued that Megalodon belonged to a completely separate genus, Ototus, within the entirely separate family Odontidae. Odontidae is now an entirely extinct family. Its members ranged across the world's oceans from the Cretaceous through the Pliocene and included several species of progressively larger predatory sharks that culminated in Megalodon. The great white, by contrast, belongs to a lineage that evolved separately, probably from an ancestor it shared with the mako. The two animals, the giant that went extinct and the predator still cruising the world's oceans today, arrived at similar tooth shapes and similar apex predator roles through separate evolutionary paths separated by tens of millions of years. The resemblance is real, the relationship is not. There is no Megalodon DNA. There never will be. Sharks are cartilaginous fish, and cartilage is among the least durable biological materials in the fossil record. It degrades quickly, and in almost every case, what survives from an extinct shark is what does not dissolve: the teeth, which are made of dentin coated in enamel, and occasionally the vertebral centra, which are calcified but not bony. From the 20 million years that Megalodon spent as an apex predator across the world's oceans, the fossil record consists primarily of teeth and a small number of vertebral columns. The genetic material that determined what Megalodon was, its physiology, its immune system, its developmental biology, its sensory systems, is gone entirely. What researchers have instead is chemistry. Tooth enamel is chemically stable over geological time in ways that organic tissue is not, and it records the biology of the animal that produced it in ways that isotope analysis can read. In 2022, a team led by Jeremy McCormick at the Max Planck Institute for Evolutionary Anthropology published results of zinc isotope analysis on Megalodon tooth enamel, comparing the signatures to those of modern sharks and other marine vertebrates from the same fossil formations. Zinc isotopes in tooth enamel track trophic position with considerable precision, because the ratio of zinc 66 to zinc 64 shifts predictably as you move up the food chain. The McCormick team's results placed Megalodon at a trophic level substantially above the great white sharks found in the same fossil deposits. Megalodon was not competing with great whites for the same prey. It was eating animals that great whites were eating, and then eating the great whites. Earlier work on oxygen isotopes in Megalodon teeth had raised a question that the zinc study reinforced. The oxygen isotope ratios in shark tooth enamel record the temperature of the water the shark inhabited during tooth formation, but they also carry a signal from the animal's own metabolic heat. The same signal researchers use to distinguish warm-blooded marine reptiles from cold-blooded ones in the fossil record. Megalodon teeth from multiple formations showed isotope signatures inconsistent with a purely ectothermic animal whose body temperature matched the surrounding water. A 2023 study on clumped isotopes in Megalodon enamel tested the question directly and found signatures consistent with body temperatures significantly warmer than the surrounding ocean, in the range of what a highly endothermic animal would produce. An animal of Megalodon's size, generating and sustaining that kind of metabolic heat, would have required a prey base of extraordinary scale to support the energy budget. What comparative genomics of living Lamniform sharks fills in is the broader biological template from which Megalodon was built. The order Lamniforms includes the great white, the mako, the salmon shark, the thresher, and several others, all of which have been sequenced in recent years as part of large-scale shark genome projects. The genomes of Lamniform sharks carry adaptations for high-performance predation that appear consistently across the order. Enhanced sensory gene families associated with electroreception and lateral line sensing. Expanded gene families related to wound healing and immune response, and metabolic gene networks associated with regional endothermy that allows these animals to hunt efficiently in cold water. Megalodon, as a member of a related but separate family within the same order, almost certainly shared many of these genomic features while having developed others independently across its 20 million year lineage. The Odontidae had been evolving separately from the Lamnidae since the Cretaceous. Whatever Megalodon's genome looked like, it was not simply the great white genome scaled up. The size question itself has been revised along with the phylogeny. For much of the 20th century, estimates placed Megalodon between 18 and 25 meters, extrapolated from tooth size using great white body proportions as a reference. When the phylogenetic revision established that Megalodon was not a scaled great white, those estimates became suspect. A 2021 study by Kenshu Shimada and colleagues used body proportion data from ododontid relatives rather than great white ratios to produce revised estimates. The new figures placed Megalodon at roughly 14 to 17 meters, still the largest macro-predatory fish in the history of life, but built on a body plan that was almost certainly different from the great white in ways the fossil record cannot fully reconstruct. Megalodon went extinct approximately 3.6 million years ago. The timing coincides with a major reorganization of marine ecosystems at the boundary of the Miocene and Pliocene, when global cooling altered ocean circulation and productivity, and when baleen whales, Megalodon's likely primary prey in its later period, underwent significant changes in distribution and species composition. The closure of the Central American Seaway around 3.5 million years ago, which severed the connection between the Atlantic and Pacific, disrupted the prey populations Megalodon depended on across both oceans simultaneously. An apex predator maintaining body temperatures well above ambient and sitting at the absolute top of the marine food chain requires an enormous throughput of energy. When the ecosystem supporting that energy budget began to collapse, there was nowhere else to go. What is left behind is 20 million years of teeth scattered across every ocean on Earth, a handful of vertebral columns, and the indirect record written in isotopes and the genomes of distantly related living relatives. The animal those fragments describe was not a great white scaled up. It was something the ocean has not seen since and may never produce again. An endothermic apex predator of enormous scale, built on a separate evolutionary lineage from the sharks we know today, running a metabolic engine hot enough to hunt in cold, deep water, and feeding at a trophic level nothing alive currently occupies. The 1843 classification put it in a box that fit the teeth but missed the animal. It took 150 years of closer looking at enamel microstructure, at isotope ratios, at the genomes of living relatives, at the chemistry of fossils nobody expected to yield any biology at all, to start seeing what was actually there. The great white is not a smaller megalodon. Megalodon was never a larger great white. They were two separate answers to the question of what it takes to be the largest predator in the sea, and only one of those answers is still alive. Subscribe. New discoveries about lost species posted every few days.